we'll get started. You can always tell that we have an alpha speaker in the room when so many people come to listen. And it is a great pleasure to introduce my good friend and colleague, Andrés Canales-Johnson. Andrés completed his PhD in this department. He's now an affiliated lecturer. He's the principal investigator in the Consciousness and Cognition group in the department. And you also hold a faculty position in your home country of Chile. Andrés' research combines various electrophysiological techniques, EEG and more invasive ECOG, to study perception and predictive coding. And rather uniquely, actually, your work is cross-species, so you combine work in humans, animals, to resolve the cortical mechanisms of large-scale neural communication. And I know this has led to some really high-profile recent publications. So no pressure, Andrés. I know from our students that you're a great communicator and lecturer, and we're really looking forward to your talk. So thank you.
Thank you very much. I promise I didn't pay Jeff for the introduction. So thank you very much for the invitation. It's a pleasure to be here where I've been for already almost 10 years with some, swaps between countries, but mainly here. And what I want to do today is to present you an alternative view, if you want, of how the brain is capable of orchestrating information for perception and predictive processing. The overview of the talk is this. So I'm going to try to go through these 4 points in around 50 to 55 minutes, hopefully. I'm going to present you first two main challenges that I, as a researcher, have been trying to solve during the last 10 years or contribute to the field during the last 10 years. The first is the challenge of studying perception, and in particular, how you can conceptualize perception not only as something that is passively acquired from the external world, but also something that is endogenously or internally created by the subject. The challenge of how to study this from a neuroscientific point of view, in particular, what type of brain dynamics is the one that we need to look at to study how the brain orchestrates information for perception and prediction, and I'm going to try to explain what do I mean by that. The main point of the talk today is trying to convince you that if you look at the non-oscillatory part of your signals, the part of the brain that doesn't have any particular rhythm, even though it looks like noise, that noise contains a lot of information for perception and prediction. And then I'm going to just try to give you examples of how that non-oscillatory activity can tell us something about perception and also something about prediction. So that's the aim of today. So I always start very simple by saying that when we study perception or the contents of perception, what we neuroscientists traditionally do is to have different instances of percepts, for example, images, in this case, cats and houses, and we record brain activity, and we try to understand, okay, what's the difference in the brain when you perceive, in this case, for example, an image of an animal versus an image of a house. I would say that this kind of approach, it expresses or it understands perception as something that is given from the media. So basically you get information that comes from the stimuli and you try to understand how the brain processes that. I call that externally driven perception because it's mainly driven by the incoming brain activity. But things are not that simple. And we know from many neuroscientists, I'm just citing two of them, my heroes here, Francisco Varela from Chile and Walter Freeman from the States, who basically present this view that perception is not only the passive processing of information from the outside world, but it's mainly, you can see it as an active process. It's not just acquisition of information, but it's a process of inference. So there is something about the internal activity of the brain that contributes to our perception. And that view has been conceptualized as the internally driven aspect of perception. An example of that are these images that you can see here. For example, during this process called perceptual ambiguity or perceptual bistability, you have images like this one that you can perceive either as two faces looking at each other or one single base, or you have the classic example of a young lady or an old lady, depending on how you experience this image. The point here is that the input, retinally speaking, is exactly the same. The thing that is alternating is your experience of the stimuli. And that is an example of how your perception can change even though you have constant or unchanging stimuli. So how do we come about in the brain? For example, if you want to understand where in the signal do I need to look to study these transitions, for example, when you experience the two phases or when you study the base, the problem is much more complicated than in the previous case because you don't have the exact onset when the perception change. There are no button presses or there are no actual changes in the stimuli that you can actually lock to the brain activity. So I'm going to show you some examples of how you can study this. So first problem, the problem of how to study the internally driven aspect of perception. The second big problem is the challenge of understanding how different brain areas are capable of integrating information, right? And there's been like a massive debate in the field of neuroscience, not only cognitive neuroscience, but basic system neuroscience, on whether oscillatory activity or non-oscillatory activity are the dynamics that we should care about. And what do I mean by that? If you look at the brain, and if you look how these brain areas, for example, if you have three brain areas here, and you record electroencephalography or electrocortical recordings from inside the brain, could be a human brain, could be an animal brain, what you can see are this very, very prominent activity that if you plot it in a spectrum, you see them as peaks. So if I depict this signal as how much energy I see in each frequency, you can see that there are these little peaks. And those peaks in the spectrum are interpreted as oscillations, so activity that fluctuates with a certain rhythm. But most of the brain activity, if you see at these peaks, around these peaks, is mainly this flat line, right? Now that flat line, it's what we traditionally know as non-oscillatory activity. You can read it as 1/f in the literature as broadband activity, but when I say non-oscillatory broadband 1/f, I'm referring to the same phenomenon, which is this straight line in the spectrum or activity that doesn't have any particular temporal peak, right? So in the brain, we always have these two types of activity going on. The third problem is, okay, I have that activity in the brain. So which of these two neural dynamics should I care about when I study communication in the brain? So what we explained that with Martin Vink in that neuron paper is that when you traditionally observe what people do, if you want to understand how two brain signals are correlated or coupled to each other for a specific manipulation, what people traditionally do is to study these oscillations. And they traditionally study them by asking the question of whether the phase of a signal it's correlated with a shift in the same of the phase at the same frequency in another signal. The problem with that is that we know that by doing this kind of analysis, you only have access to the oscillatory part of the signal, not to the flat line that I just showed you. So there are other accounts, for example, instead of looking at the linear aspect of the connectivity, you can look at the nonlinear aspect of the connectivity, which is basically how different patterns of activity in one signal are related to different patterns in another signal, potentially in the absence of any kind of linear connectivity or linear relation. Why is this important? Because we know that this 1/f activity that I just showed you, this flat line, could be generated by interaction between different frequencies. So if you have energy from one frequency transferred to another frequency, you might generate activity that doesn't look like an oscillation. But there are still interactions that are meaningful. So just as an example, this is a classic idea of studying connectivity from a linear point of view. You map an oscillation, you extract an oscillatory phenomenon from your signal, from the other signal, and you ask how coordinated in phase these signals are. And the traditional example that I make is that this is like jumping together. So if two groups of people jumping at the same time or jumping with the same delay, and they preserve the rhythm, well, we can ask the question how coordinated these two people are in their jumping. But to have an intuition of what do I mean by this nonlinear activity or nonlinear connectivity, imagine the case, this is an example of one metric that can capture that. That is called WSMI, but that doesn't matter. What matters is the concept. The concept is that you can have coordination between signals even in the absence of linear relationships. And the metaphor here is dancing tango. So imagine that we're dancing tango. So each of the partners in the couple might be doing something different at one time, but in the overall dynamics of the dance, we can say that they're still coordinated. So they don't need to be doing the same thing at the same time to be coordinated over time. So this is a way of conceptualizing a nonlinear interaction, because you can have patterns that co-occur over time, but they still have a relationship between them. And there are metrics that capture that. So, for example, in this 2019 study, what we did with Laura, who was a part 3 here, by the way, what we did was simulating brain activity in 2 areas. And then what we did was to manipulate the interactions between these 2 areas, either by some boring linear equation that correlates these 2 signals, or incrementing the nonlinearity. So the coupling was more and more complicated as a function of different physical systems. And what we tested was the hypothesis that this linear or oscillatory connectivity metric should be better at capturing linear regimes, while the nonlinear one should be better at capturing nonlinear regimes. And what we found was exactly that. So if you care about the oscillatory connectivity, so the jumping together captures really nicely linear interactions, but the dancing tango, so the nonlinear connectivity metric, captures very nicely highly nonlinear interactions. Interactions that you shouldn't even be able to see by eye. So they look like noise between 2 signals, but we knew that there was an interaction because we manipulated it in the model. So this paper basically showed that there are metrics, in particular these metrics that are derived from information theory, that can capture these nonlinear interactions really well. So a lot of problems. So now I'm going to try to give you examples with data from humans and some other species on how this non-oscillatory activity is actually the one that conveys or encodes most of the information in the first part, perception. So the question for the next few slides is, what role these different dynamics play in the distributed representation of this internally driven aspect of perception? One example is, again, bistable perception. And in this study in 2020, what we did was to present a bistable stimuli that is not a visual one, but is an auditory one. So you can hear these 2 tones separated by a gap. You hear them like bitty beep, bitty beep, bitty beep, bitty beep, bitty beep. They call them streaming. And when you experience this for around a minute, you can start perceiving the tones as one single object, like a galloping rhythm, or it alternates to another version of the same stimuli that you experience as 2 objects, the upper frequency in the background and the lower frequency in the foreground. So you alternate between one auditory object and 2 auditory objects, one auditory object, 2 auditory objects. And what we did was ask, with EEG, 33 participants that basically pressed a button every time that they experienced a change between 1 and 2, and another button when they experienced a change between 2 and 1. And we recorded brain activity. And our hypothesis, for theoretical reasons that I'm not going to mention, but they are related to consciousness, we decided, or we hypothesized, that these nonlinear connectivity metrics would be much better at capturing the differences between these percepts. Remember that the stimuli does not change. The only thing that changes is your perception of the grouping. So you either experience the stimuli as one object or 2, but the stream is always constant. That's why it's bistable. And what we found was that if we compare the switches between two objects to one object, or one object to two, 0 here represents the moment where participants press the button. And what I'm actually showing you here is how much of this nonlinear connectivity is expressed between frontal and parietal regions. And you can see that almost 1.5 seconds before they press the button, you see an increase in this measure of nonlinear activity when they transition to one object, and a decrease in the same metric when they transition to two objects. So basically, with this information theory metric, you can distinguish between the two types of percepts. We also had one patient that had intracortical recordings in a superior parietal lobe and some parts of the midfrontal gyrus and the orbitofrontal cortex, and we observe exactly the same phenomenon. So integration goes up, so basically the nonlinear metric goes up just before they press the button when they experience the integrated percept, and it goes down when they experience the differentiated percept. We also had a control in which we manipulated the sound. So instead of being the perception internally driven, we actually changed the sound. And when you make a change in the sound, this marker doesn't do anything. So you cannot discriminate the change in the stimuli when the stimuli changes. The metric is only sensitive to the change when perception changes, not when the stimuli changes. There is another metric that tracks that, of course, but you can have a look at the paper. Of course, people that work in oscillations, that are many, ask me, well, sure, but if you run your traditional oscillatory metrics, what happens? Well, nothing happens. So this is the contrast with the PLI metric, so the metric of oscillatory connectivity. It cannot distinguish the percepts in the EEG patients, and also it cannot do it in the intracortical patients. So there is something about the nonlinear relationships between the signals that is better captured by this information theory metric that the oscillatory ones cannot do. So since I come from a field that is the field of consciousness, I encountered certain people that will say, okay, that's really cool, but they're pressing buttons. And if you're pressing buttons to report, your neural marker that separates the percept might be contaminated by the decision making or the post-perceptual processes, as they call it. How do you know that the marker, that this change in non-linear connectivity is exclusively related to perception and not to post-perceptual processes like decision making, introspection? So that was my first postdoc that I did in Amsterdam. And then we came out with this task that is a visual task. And we have an equivalent of these two types of percepts. So you have these gray things that you can show on the screen, that there are these lines that look very weird, but they're moving on the screen. And the interesting thing is that we manipulate the frequency at which they move, so you can see them as moving up on the screen, again, one integrated percept, or you can see these two gratings moving apart. So you again change from one object to two objects. So this is the visual version of what we had auditorily. We can have one percept or two. And again, this is internally driven, so the stimuli is constantly moving, but you experience it as moving up or two objects moving apart. But the trick here for answering these people was, okay, let's measure the EEG, but also let's measure eye tracking. And when you measure eye tracking, you can measure what is called optokinetic nystagmus, which are very, 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 very small movements of the eyes that the participant cannot even tell that he's moving his eyes. But you can use these movements to decode or to infer the perceptual button. So we train a classifier in the eye data to predict the response, and we were happy about this. We applied the same model to a condition where participants were not pressing buttons at all. They were just passively looking at the bistable stimuli, and they were not pressing buttons. So we tried to do some kind of a mind reading in a very lame way, which is with this gradient. But anyways, so just to make the story short, you can train your classifier really well using the eye tracking data to predict the button presses with like 85% accuracy over chance. And then when we were happy with this, we just apply the same method to the no report condition, and you could also decode the two types of percepts in the absence of response. So now we have a method that we can use to kind of read or mind read whether they're seeing one object or two without the responses. And we analyze the data in the same way as before. We had a bit better measure of information, nonlinear information, that is directional. So the only difference with the previous metric that I showed you is that with this metric you can tell whether the information flow goes from one part of the signal to another or the other way around, meaning that you can infer lags between the information flow. This measure is called transfer entropy. And we were interested in this because, again, the theories of consciousness claim that the important information should be top-down. So we should see mainly effects in transfer entropy from frontal electrodes to back rather than from back to front. And we also applied a metric that is being used in the field that is called Lempel-Zib that helps you to understand how much information or how much diversity of information you have in a signal. And it's an indicator of how differentiated the signal is. And the prediction was that when you're experiencing these two objects instead of one, you should see more differentiation of information in this two-object perception versus one. So, what happens in the report condition? So in the report condition, 0 here represents the moment where they press that they were experiencing one object, and here that they were experiencing two objects. When you compare the transfer entropy from frontal to back electrodes, you see a very nice effect like 1.5 seconds before they press the button in magenta here, with a lag of 100 milliseconds and almost a lag of 200 milliseconds. That tells you that there is more information flowing from front to back when you see one object than when you see two. So this is similar to the result that I showed you with the auditory case. So you have more nonlinear connectivity, in this case directional, between front to back when they're experiencing one versus two. And we have nothing from back to front. So basically, if you want, the internally driven perception is mainly driven by this top-down processing rather than just bottom-up. And interestingly, we observed that the complexity metric was higher for the percept when it was experienced as two objects as when it was compared as one object. So you have more differentiation of information when you say, okay, I'm seeing two objects versus when I'm seeing one. So far, that replicated in a bit more sophisticated way the auditory case. But then what happens with the mind reading? What happens when I lock the activity to the eye movements? Well, something similar happens. We still have the effect from front to back. So we still see more information flowing from front to back when they experience one versus two. There's still not much information bottom up. But critically, and this is why my reviewer tree almost had a psychotic break, we couldn't find a difference in the differentiation metric. So now, this measure of compression doesn't tell you anything about the identity of the person. And this is important in the field because there are a lot of debates about whether this Lempel-Zib complexity is a marker of the contents of perception. So you can use it as an indicator of how percepts change in your mind. We couldn't find any difference in the no report case. So what is the conclusion of all of this? The conclusion is that if we care about the neural marker of perception and not post-perceptual processing, well, it seems that the one that holds is the information transferred from front to back because we find it in the report case and also in the no report case. But the measure of differentiation, since we find it only in the report condition and not in the no report condition, of course the absence of evidence is not evidence for absence, but it suggests that this metric could be more related to the act of reporting rather than the act of perceiving. They're still unhappy, all the people in my field. We haven't solved the problem, but that's another story. So this last example, before changing gears, is an example of a very, very basic phenomena that happens in olfaction. So if you are a bit skeptical about, okay, Andres, that's nice, your measures are cool, but that's EEG, so who knows what's going on? Well, what we did here was try to show whether these metrics were more sensitive to discriminate odorants. So we did it in fish. So this is a trout. We had 16 trouts with electrodes in the olfactory bulb and 2 areas of the brain that encode information about odorants. And then here what you see is that this is the moment in the electro-olfactory where you present the odorants. The trouts, by the way, are in a tank with water, and they're restrained, but they can move their tail, and then we make the water flow backwards. So the trout, it's like a swimming simulator. The trout probably experiences the water flowing, and then you puff the odorant, and the moment that you puff the odorant in water, you see this beautiful oscillation that has a peak around 7 hertz, and it lasts forever. It's a true oscillation. It lasts 10, 15, 20 seconds. So my colleagues in Chile were saying, if we see oscillatory activity, the mantra, if we see oscillatory activity between 2 areas, the connectivity has to be oscillatory. So the linear metrics should hold. So the first thing I did is that we tried this linear connectivity metric, and it performed really well at separating 3 types of odorants against the control. Look at the scale, 0.2. But it didn't discriminate really well the 3 types of odorants by themselves. When you apply the same, in the same frequency band, the nonlinear one, now you can separate odorants from each other and odorants from control. And you can see it here that the scale is almost like 3 standard deviations above the baseline. So the nonlinear connectivity metric, even in a case where you have electrodes and you have a very clean, clear oscillation, it's better at capturing the odorants than the linear one. So far, what I've said is that neural signals are complicated, so they're usually a mixture of oscillatory and non-oscillatory activity. That if you want to study brain communication between areas, you should consider nonlinear metrics because they capture better aspects of the dynamics that is not necessarily linear or proportional, and that these metrics are very good at capturing perceptual information across species and also modalities, if you want, vision, audition, and olfaction. Wow, I'm doing good with it. Okay, so now the final part of the talk is related to the aspect of perception that is inference itself. So by that I mean all the accounts on predictive processing and how perception can be seen almost like a hypothesis testing of the world rather than just passive extraction of information. And I'm going to give you some examples of how we can solve this question. So now again, what role this distinct dynamics play in the large-scale integration of predictive information. And we got a paper yesterday accepted, so it was really good for the talk. That so with Martin Vink, again, what we're basically claiming is that the inference of the stimuli depends on these non-oscillatory transients. So in the field of predictive coding, there's a huge debate on whether the oscillatory part of the signal is the one that encodes communication of errors. And there are some papers showing that that's the case. What we're claiming is that we should care more about this initial swipe, this initial transient, because it could contain all the non-linear interaction that you need for the swipe of errors towards the other areas. So I'm going to show you some evidence of that claim, and then I'm going to show you some evidence of how we should also care about the interactions between areas. So the story goes like this. I was bored to death during the pandemic. I was 2021. And then I started reading a lot about this predictive processing. And I wanted to, in the literature, as an innocent man, reading, okay, what are the neural markers of this prediction error encoding, right? And then I found that if you do a search, you can find that these prediction errors are basically encoded in every single frequency band that you can think of. So you can find papers that they show that is in gamma, in beta, in theta, in alpha, almost everywhere. So I asked myself, okay, there are probably two explanations for this. So we have exactly the same, we have a lot of independent mechanisms that explain the same phenomena, or it could be the case that this is again a broadband effect, right? An effect that is in the broadband part of the signal and not in the rhythms. And by then I met Kai Miller, and he was doing a lot of work in how to separate from the signal this broadband activity from rhythms. So it was, okay, cool. Now I can use this method to extract rhythmic activity and non-rhythmic activity and see which of the two phenomena are explaining more the prediction error. Now this broadband activity, when you look at it in the LFPs, it's nothing fancy. Basically, it reflects the how neurons respond to asynchronously to a stimuli. So it's not an oscillation. It's basically a noise distribution of spikes that you can see as this 1/f effect that doesn't have any particular peak. So when I talk about broadband, I'm talking about how people call this non-oscillatory activity in the LFP world, people that do intracortical recordings. Mike says yes because he works on this. So if you have that's, by the way, that's not a Pokemon. It's a marmoset. A marmoset is a little monkey. And so I started analyzing data that was in the website, and I contacted the person who did the experiments, and she sent me more data. But basically what the task was you present repeated stimuli, and then you have a deviant tone. So beep, 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 boop, 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 boop. And then if you calculate the difference between your standard tones, the repeated one, and the deviant tone, you get what is called, the difference is what is called the prediction error. Because the brain is creating a model of the same stimulus, predicting that the same thing is going to come over and over again, and suddenly you have a deviation. So the difference between the two tones in the neural activity is reflected as this huge effect that goes almost from 10 hertz to 250 hertz. So the first thing that I say that how on earth this can be an oscillation. Oscillations are things that have a fixed frequency. Here we have an effect that almost has 250 hertz. So how can we separate the broadband from the oscillation? There is a method that Kai invented that is very simple when you go through it. But basically, if you take trials of your two types of tones, you create a spectrum, and then you do a PCA. The interesting thing is that in the LFPs, if you check how the first component of your PCA look like, it looks like this. It looks flat, meaning that all the frequencies are equally or roughly equally represented. They have exactly the same amplitude. So this component is the broadband component, or the non-oscillatory component. While if you take a look at the second or third component, you usually have peaks. For example, this second blue peak is a rhythmic one because it has a peak in alpha, around 10 hertz. And this third one has a peak in delta because it peaks around 2-3 hertz. So data-drivenly, you can take the data and then reconstruct your time series only with the broadband or only with the rhythmic. So you have a nice way of taking the data that is a mixture of oscillatory and non-oscillatory activity and separate what is what and reconstruct your time series. And when you do that, and you compare your deviant tones, 0 is the moment when you present the tone, your deviant tones, again, your standard tones, the broadband activity, so the activity that doesn't have any rhythm, is the one that reconstructs all the peaks and the difference between the two tones, so the prediction error. If you do the same with the rhythmic ones, you see absolutely nothing. So the rhythmic activity extracted from the data does not contain any information about the prediction error. It's only the broadband activity that contains. And if you think about it, again, this broadband activity reflects spikes. It's like a shadow of the spikes. So what you're actually measuring there with this first component of the LFP is the local spiking activity. So it makes sense that most of the information is contained on that broadband rather than in the oscillations. To me, maybe not to you. And we had two other monkeys, and we actually saw the same effect. So the broadband separates very nicely the deviant and the standards, but not the oscillatory activity. So the summary of this is that the non-oscillatory or broadband component encoded most of the prediction error information in the auditory cortex. If we do this method and you extract whatever oscillatory components you have in the data, they couldn't separate the two types of tones. And for the gamma people out there, I couldn't find any kind of gamma peak. So I'm sorry. They know who they are. Okay, good. So the next part is about how these transients can contain information for communication, right? So when I talk about transients, I'm basically talking about the first part of the response, that is the non-oscillatory part. And we suspected that this initial transient should be, or should have, or contain most of the information that allows you to communicate the prediction errors from one area to the next area. There are indications that these transients, if you look, for example, at ERPs, and if you have any kind of modulation, for example, when you make people to imagine things, these transients in the form of ERP can be modulated by this top-down processing. So even early potentials or early transient can be influenced by areas that are not necessarily around. It could have top-down processing. So that suggests that there is information in these transients for communication. Another source of evidence is that these transients can contain information, for example, for decoding expressions in faces, almost at 4.4 milliseconds. So this is a study that we had an equipment that is called a tachistoscope, that you can present stimuli as brief as 2 microseconds, so 0.002 milliseconds. If you present stimuli at 4.4 milliseconds, the brain can immediately tell whether there is an emotional valence in your stimuli. And these transients are very, very clear. Well, there is one there at 4.4, but at 6 milliseconds, you already have them formed. So again, there could be something in these transients that is relevant for the encoding of inference. So we did it again in the auditory cortex of these marmosets, and here the question was, okay, do these non-oscillatory transients encode certain kind of recurrent interaction? So can we use a metric that tell us whether these transients are encoding distributed activity across the brain? And we have a candidate metric that is called synergy. Synergy sounds like esoteric, but it's not. So synergy could be a reflection of the recurrent processing. What is synergy? Well, in the context of this study, and in information theory, if you take two signals, for example, a signal from the temporal cortex and a signal from the frontal cortex, and you estimate how much information you have on a trial-by-trial basis, you have something that is called redundancy, which is the idea that these two signals have something in common, or they're sharing some information that is the same. So you don't gain more information about the prediction error contrast if you look at any of the signals separately. There is this redundant effect. Now, there is something called synergy that is a superadditive effect. And the idea is that if you now combine these two signals, you gain information about the contrast, in this case, standard or deviant, only when you take them together, right? So it's the idea that the sum is more than, how is it, Lupi? The whole is more than the sum of the parts. I always get it wrong. Yes. But for this specific task, what we wanted to know, and we want to use the synergy as a metric of distributed processing, right? And hopefully to show that these synergistic interactions convey information about the recurrent process. So this synergy might potentially indicate the presence of recurrent processing. And we also wanted to know, if we found synergy, what was driving the synergy in the cortex, right? So we did this study again. We have 3 marmosets for the oddball task and another 2 marmosets for a more complicated global local task that Tristan invented almost 20 years ago. 13, thank you. And then you can run this task, and then you can compute again your favorite transients. So the broadband, the non-oscillatory activity, and we can also look at the ERPs. So we can check whether these transients actually convey synergistic information. So this is an example of all the electrodes in the marmoset brain that showed a prediction error effect. If you see, the ARPs are very distributed, but the broadband, so this non-oscillatory 1/f thing that I've been talking about for the last hour, is very localized, mainly in the auditory cortex and some part of the frontal cortex, because it reflects local spiking. So what you can do is just to compare, to contrast these signals. And now the question is, okay, if we check how much redundant information or synergistic information you have in one area, do you see any synergy? That was the first question. And this is, for example, what happens in the auditory cortex of these marmosets. Here what you see is the difference, or the effect size of the difference between the two types of tones, thunder and D-bass. And what we're plotting here is the redundancy and the synergy as a function of time. Every time that you see red, what you have to read is that these time points contain exactly the same information about your contrast. So they convey the same information about the prediction error around these peaks. So that's why you see red there. Peaks are time points against themselves. But if now you combine what happens in the peak against what happens in the bump of the signal, now you get blue. And blue for this metric means synergy. What basically means is that whatever process is going on at this moment in time, when you combine it with whatever process happens at this moment in time, they give you superadditive information. So more information when they are taken together. So you tell better whether your stimulus is standard or deviant when you take these two time points together and if you take them separately. So there are synergy even in local circuits, if you want, even in the auditory cortex. But our question was, what happens between cortex? So now if you test the effect in the temporal cortex against what happens in the front, you also see synergistic effects. You see redundancy, but also the important bit is that you see synergy. So now there is the synergy here, and you also see it in the broadband. So in the two types of transients, for the ARPs and also for the broadband effect, you see it at the end. What this means is that the prediction errors are not necessarily what the theory predicts. For example, hierarchical predictive coding would say that you have a copy of your prediction error and you have to move it from the next area almost intact. Here, this synergistic effect, what I'm actually telling you is that the encoding of these errors is a distributed effect across the network, because the synergy is something that only comes out when you have the two signals together. You say that sounds beautiful, but tell me something about the brain. Okay, so the next question was, okay, what is driving these synergistic effects anatomically? Right? Unfortunately, we cannot cut brains yet, but you can use simulations. So what we did, and this is a very straightforward simulation, I promise, we used these brain-constrained neural networks that are very nice because they respect the biophysics of the brain at different scales. And what we did was simulate 6 areas that are important for the marmoset brain. And we trained these networks from A1 till motor. We created 2 architectures. One architecture that only moves information forward, so it doesn't have any kind of recurrent loop. And an architecture that has recurrent loops. So it has recurrency within areas and also recurrency between areas. And we trained these networks to identify or to distinguish between the two types of tones, and we generated in silico time series. And on those time series, we computed the synergy and the redundancy. And the prediction was very simple. The prediction was that only the architecture that has recurrency should show synergy. Because if this synergy is actually related to loops and recurrency, it should be only in this architecture, but not in the architecture that is only feed forward. And the result is, well, if you take the feed-forward network, you generate time series in the temporal and the frontal, and you compute the co-information between these two. You see redundancy, but you don't see synergy. If you have an architecture that respects the loops that you see in the marmoset brain, you generate activity like the one that looks in the temporal and the frontal activity. When you compute the co-information, you see redundancy, but critically you see the synergy. So this is important because at least we cannot cut anything again, but at least we can reproduce the effect that the experimental results show, and we can further show that the only architecture that generates the synergy is the architecture with recurrence. 5 minutes? Good. So we're doing this now with marmoset. This is not a Pokemon again. This is a marmoset that has wireless echo. You have 128 electrodes, but you don't have cables. So now what we're trying to do is to study the same phenomena, but while the marmoset is actually able to move around. And then you can study how these synergistic interactions are modulated by the state of the sensory motor loop. I don't have anything to show you about this, but I think it was cool to show you that. So if you invite me in 2 more years, maybe I can say something about it. So the conclusion about this part is that this synergistic information was encoded by the transients, so the non-oscillatory activity, and it was observed between regions. And also with this brain constraint neural networks, you can show that the synergy emerges only when you have recurrent links between the areas. Now, to finish up, I want to show you two more slides that we have now. The obvious question to be that to what extent this oscillatory activity and this non-oscillatory activity convey synergy or redundancy, right? And we use a system where you can very clearly separate the broadband from the oscillations, and where people believe that these oscillations should convey synergy because they should be important for communication. And this is the visual system. And this is data from Dora Hermes from the Mayo Clinic. And what she showed was that if you have stimuli that have a lot of gratings, that's the only kind of stimuli that can show you a gamma oscillation in V1. The only type of stimuli that can show you a gamma oscillation. The moment that you show something that is not a grating, like noise or phases, the response is immediately non-oscillatory or broadband. So you only get a narrow band gamma oscillation when you have this type of stimuli. So the question is, okay, so if the theories that claim that this gamma oscillation should convey meaningful information for integrity process, then the gamma should have more synergy than the broadband. So, long story short, we have 2 patients that have electrodes in V1, ECOG, V1, V2, V3. And then what we did was to estimate the difference between these gratings against stimuli that are not grating but are more noisy, so they evoke more broadband. You do the same method, and now with the decoupling, you can have the broadband recovered or only the narrow band gamma. And the question is, now when you separate broadband from gamma, which one has more redundancy or more synergy? We expected that the broadband, so the non-oscillatory thing, should have more synergy, and the other thing should be only redundant, the other thing. So the oscillation should have more redundancy. And that's exactly what happened. So we can separate very nicely the broadband effect from the narrow band gamma in V1. When you test only for the broadband, the interactions between V1 and V2, but it also works on V1, V3, most of the synergy, you see redundancy, but there is synergy in the broadband. And the narrow band gamma, when you test for the communication, there is no synergy at all. There is mainly redundancy. So this will upset even more people, because it would basically claim now that, okay, this gamma oscillation is just some kind of signal that only convey redundant stuff. So you don't gain integrity process as a function of how you move in the hierarchy. It's just a copy that moves and moves around. The broadband, or again the transient, is the one that contains, if you want, the recurrency between the areas. So that's what I just said. So to conclude, so I hope today I have convinced you, even partially, that if you do electrophysiology in any form, you should care maybe a bit more about the non-oscillatory part of your signal, because that oscillatory part of your signal, believe it or not, it's very meaningful for understanding perception when the perception is not necessarily externally driven, but also internally driven. It contains information about perceptual inference. So if you apply methods that decompose the information, you see that most of the recurrent activity is in that non-oscillatory part of the signal. And we have methods to tease apart what is oscillatory from what is not. So thank you very much. I want to, before that, thank all the people because I haven't done an experiment in like 10 years. So, well, of course, Tristan, that has helped me and supported me for the last forever. Robin is, that I do all the information theory with him. Max Gergiani from Goldsmith, that we do the simulations. From the Netherlands when I did my postdoc. Simon van Gaal for all the perceptual study that I showed in the beginning. Also Johannes Farnford. From the US, all the ECOG from patients, from Kai, Dora, and Mike that he's visiting. From Japan, all the marmoset stuff with Misako Komatsu from the Riken. All the theoretical work and mice work that I didn't show because it was too much with Martin Denk and Sam. And the trouts, of course, from the Chileans. So thank you very much.
Well, you'll agree with me, that was an extraordinary talk. Thank you, Andrew. That was amazing. Questions? Yeah.
Thank you. It was really good talk. Maybe start from the end. So, do you think this gamma oscillations are just the same transient that is again and again and again, or
so you could, I mean, you could think in general as oscillations as what we're postulating in that TICS paper is that these oscillations might be good for keeping information stable over time. So if you want to keep a representation in a specific area for a period, you need something that has like a tight excitatory inhibitory balance. So oscillations could be perfect for maintaining information ongoing in an area, but not necessarily. What we're claiming is that they are not the ones that carry the information for communication towards another area. So it could be good that this gamma work for plasticity within an area. Bob Singer is postulating that now, for example. Or that the gamma oscillation in V1 is a signal that you need to maintain the information online. But what we're claiming is that it's not a carrier of anything that communicates that information towards another place.
So you don't think there is a relationship between the transients and oscillations?
That's a good question. I would say no. I would say that every time that you stimulate the brain, if you have an ongoing brain doing oscillatory things, every time that you have a stimulation, you have a disruption in the oscillatory activity by the transient. So the transient should be by itself seen as the disruptor that contains or ignites the information towards another area. But probably they don't, that's an open question, but I would say out of my head now that they should be independent processes.
Other questions? Paul?
Of course, very naive questions. I'll stick for one
Me too.
for now anyway. So you said that the absence of synergy meant that all the oscillations were doing moving, copying information from place to place. But that sounds a lot like the role that people ascribe to oscillations, right? They transmit information between different brain areas, as in the classic claim that binding in the visual system relies on that. So can you explain that to me? Why do you expect to see this synergy?
Yes.
Don't you just want copies?
Yeah, so if you think about the visual hierarchy, and if you think about the traditional model where information is being processed and then integrated from V1 to V2 and from V2 to V3 and V3 to V4, what I should expect is that the representation, it gets more and more complex as a function of passing area. So you should have information that is more integrated, right? Why would I see a copy of the same information that is being processed in V1 until V4? So where is the integration part? And I'm seeing the synergy as a way of signifying or signaling integration. At least because it reflects recurrency. If it reflects recurrency, you might be, I don't know, integrating borders with shapes and shape with color, etc., etc. That's one way of answering. The other way of answering goes from the predictive processing or the hierarchical predictive processing theory, that these results kind of go against it, because what these prediction errors you should traditionally imply is that the prediction error is transmitted from one area to another. So it should be preserved across the hierarchy. But what we're showing with this synergy is that in order to have better decodability of the tones, you need the two areas working together. So it speaks more of a distributed predictive coding than just like a serial or hierarchical. I don't know if I'm answering anything, but
I don't think I answered.
yeah, exactly. But so what we're trying to do now for real with Martin, it's to, in mice, what we're trying to do, it's working so far, is to disrupt with optogenetics. So basically with optostop, you can optotag interneurons in V1, and then if you selectively cancel or optotag the transient, the prediction would be that you immediately suppress the synergy across the hierarchy. But if you optotag the later part of the signal where the oscillation is, then the communication should not be disrupted. So that would be, like, to me, the ultimate way of demonstrating that these oscillations are not conveying any information for the feed-forward swipe, but the first part, if you try to opto-kill the effect. So again, maybe in a year.
Yeah. I mean, so for neuroscientists out there, measuring oscillatory rhythmic behavior in the brain rather is fruitless, because there's no information there, and perhaps it's a futile endeavor. Are you suggesting the information then is the non-oscillatory activity? The question, I guess from my point of view, is the biological flesh of the signal. You say it's spiking, but spiking means many things, right? It could be a change in the balance of excitation inhibition.
Yeah.
I mean, what exactly do you mean by this non-oscillatory signal?
So that's an excellent question, and you can have different flavors. So for example, you can have changes in non-oscillatory activity when you have a movement of the 1/f up or down, but you can also have a change in the axis. And these two processes, or basically you can have an increase in the latest part of the 1/f, they all reflect different things. So one could be excitation inhibition balance.
Mm-hmm.
You can have changes in the time constants as well that can reflect the changes in the offset. You can have, well, I think these two are the most obvious ones. You can have changes in synchrony. So you can have a few neurons that are doing one thing at the same time, and then you can have an elevation in one part of the signal, so one part of the 1/f, but not in the entire thing. So there are many ways of changing that 1/f. There is no fixed one. The excitatory inhibitory balance is one model, but that's something that it will be also interesting to investigate which of these different ways, which of the different flavors of 1/f or mechanisms underlying the 1/f are the ones providing synergy or redundancy, for example.
Tristan.
So the part where we think about the networks that are doing oscillatory work, and they exist and they're all over the place. If you see them like in Dora's one or other places, what is the relationship at the neuronal network level with the oscillatory information we see, the non-oscillatory signal? Is it similar? Do they have a shared neural network? Are the same neurons, but they are dancing and jumping at the same time?
I don't know.
I'm asking you for you to speculate.
Speculate. Okay.
I wasn't asking for a real question.
Okay, good. So if I may, I think what I've learned in the last 10 years is that I think we need to go back to the most basic systems first to try to deambiguate what is obvious, and then to start building up. So I think at the moment maybe we're not I am not in the position of trying to generalize about networks, but if you push me, I would say that most of the brain is hardly oscillatory when you have a task. For example, this alpha activity is something that you only see it when you close your eyes. It's maybe in the back of the head. Gamma oscillations are a very, very rare event. They only happen in V1, V2 olfactory bulb, and that's it, with very, very specific type of stimuli. They are not even there when we're observing natural images or natural phenomena. So I would say that the oscillations are the exception. The point is that we have been fixated with this idea that the oscillations carry information because, not because scientists are dumb, but because they're extremely obvious in the signal. You can see them by eye. And you have methods that decompose your signal into these oscillations very simply, like a Fourier transform. So the most obvious thing to do if you have a hammer is treat everything as a nail in this case. But most of the activity in the brain is non-rhythmic. If you see a spectrum, the peaks are very restricted in frequency. So probably these macroscopic networks are mainly composed of activity that is leaking from one frequency to another, and you have these beautiful cross-frequency interactions that manifest as non-oscillatory activity. I don't know if I answer anything.
At the back.
Thanks for the very clear talk, Andrés. Does this transient have a link to, for example, gamma bursts? Because that has been sort of a new movement of trying to look at single triad gamma bursts and seeing these gaps in between. So are these sort of nonlinear methods capturing more of that because they are bursty in nature and not really rhythmic?
Yeah, in principle, they should. Now, these bursts, I'm a bit iffy about the bursts, not because I don't think they can happen, but then the question to me is more fundamental, is how many cycles you need to have in a signal to consider that meaningful for cognitive function, and also to consider this as an oscillation. How many cycles? 2, 1? Some people think that they call theta oscillations things that have one cycle because the power increases at that frequency for 200 milliseconds. They call that an oscillation, but is that an oscillation? Second, if it lasts so briefly, does it have any kind of functional implication? I don't know.
There is evidence that the bursts can be a bit synchronous across different electrodes.
Yes.
And if you quantify it, then yeah, there are different ways of quantifying the bursts.
Yeah.
And then there are, so I'm just trying to link these two sides of the story, basically.
Yeah, I would call them bursts, but I'm not sure if I'm sold with the idea of call them like oscillatory bursts. Maybe they are just increases in power at a very, very narrow band frequency restricted over time. Because you're applying a method that transforms your signal into a sinusoid, you call them oscillations. But I don't know, again, how many cycles do you need to call something an oscillatory phenomenon? Probably more than one, that's my opinion.
Any other questions?
Can I just follow
Yeah.
up on that? So one way to look at it would be to see if you have like the information-rich period, if it has a certain length.
Yeah.
When we use Fourier transform, that it will look as if it is a burst of a specific frequency that fits to the period. Do you see something like that the length of the informative period, the synergetic period, has characteristic? So there's a certain length.
Yeah, I would go about, if you could show me that these brief bursts, the entropy in those bursts is enough, or it has a correlation with the entropy of this burst in another channel, and that coordination has some correlation against behavior, even if you show me that it's one cycle, I would be more convinced, but probably...
No, I meant on the other way. So
Yeah,
when you see
with the length, yeah.
Yeah, the length of
Yeah.
the duration that you find in
That's an empirical question. That would be good to test. The other skepticism that I have is that entropy-wise, so disorder-wise, oscillations have very, very low entropy. So if I ask you to count, okay, I'm gonna count to 3, and then you're gonna say the next number. 1, 2, 3. Now I'm gonna count until 3, and the next you're gonna tell me your name. 1, 2, 3. Where there is more information? In the first case or in the second case? Intuitively. Where? In the second case, right? So activity, they're all saying 4. So basically the entropy of that, all of you telling me 4, is very, very, very low. So why would the brain care to communicate complex information all the world about these oscillations that have low entropy? It doesn't, to me, it doesn't make any sense.
John.
My question is main about terminology. So you say gamma is rarely seen in the brain, but if you read papers on human ecog, by far the most popular measure is high frequency power, sometimes divided into high gamma and low gamma and so on. But are you saying this is really better thought of as a broadband?
Yeah, exactly. So the high gamma, low gamma, it's a terminology that it's bad. So there is a paper in 2012 from Jean-Philippe Lachaud and a lot of people at the ECOG that they decided to call it high frequency activity, which is better than high frequency gamma, because every time that you hear gamma, you think of an oscillation. So then there was Kyle Miller saying, look, if you decide to cut your gamma from 30 or 40 hertz up, I can show you that if you do a decomposition like the one I show you, the effect goes until 1 hertz. So it's truly a broadband phenomenon. So I think that the distinction is meaningful in the sense that if you speak about broadband, you're truly thinking about arrhythmic spiking. While if you use the word high frequency gamma, you're cutting that broadband into a period, and you're also implying that there is some kind of oscillatory stuff there. But again, it's 200 hertz, 300 hertz amplitude. What's the meaning of even defining the phase of something of 300 hertz? Yeah.
Very thought-provoking. Good. Okay, look.
Okay.
Now, there's any other points you want to express? Well, then it just leaves me to thank Andrés for a really wonderful talk. It was superb. So, yeah, thank you.
Thanks, Andrés.
Thank you.